or less. And the title is A Breakdown in Metabolic Reprogramming Causes the Microglia Dysfunction in Alzheimer's Disease. So first, I will go in a brief overview of what they did in the experiment, so you can follow more easily the paper. So they went from the in vitro setting to an in vivo setting. In the in vitro setting, they used the primary microglia cells, and they treated them with A beta peptide, which, if you don't know, it's one of the main particles that it's accumulating in the Alzheimer's brain. And then they treat it with different forms: the monomeric A beta, the oligomeric A beta, or the fibrillary A beta, which is actually in the progression of the disease, the A beta peptide is forming the oligomers and finally the fibers that will in the end be deposited in the brain, causing the disease progression, or being one of the main causes of the disease. So then their readouts is C-course and for the metabolic analysis, mRNA and protein analysis, and also they did the phagocytic assay. For the in vivo model, they used the Flatakine GFP 5 times PAD mouse model, and there they did two invasive techniques. The one is the intracerebroventricular injection of the oligomeric A beta, and the other is the intraperitoneal injection of interferon gamma. I will go through these different experiments when time comes, but just for you to have an overview. And then they analyze through histology, RNA sequencing, in vivo imaging, and finally behavior. Going through directly through the experiment, so they established the microglia primary culture, and they treated the cells with different forms of the A beta. And what they see is that in the exposure of any of the A beta, the cells become activated. They get this deramified morphology, as you can see here. I think I can also use this kind of pointer. Yes. So you can see here that they become deramified regardless of which type of peptide they are being exposed to. They did a functional assay with the microspheres. So this is a phagocytosis assay. They put these microspheres into the culture, and they see how much of these microspheres are internalized in the microglia. And what they saw that was during the exposure to the A beta, here there is a high internalization of the microsphere, suggesting a phagocytic cell. They compare it with LPS stimulation in the cells, and they actually find that there's a similar response to the LPS when exposed to the A beta peptides. They went further. They did some Western blot analysis for pro-inflammatory cytokines, and then so here the pro-IL-1 beta is getting upregulated, and this was also shown in the RNA level. TNF-alpha was also high, again mimicking an LPS, or at least being similar to an LPS stimulation. The anti-inflammatory cytokine is interleukin 10, and the expression of the interleukin-1 receptor A is actually not differentially regulated. So from this experiment, they came to the conclusion that this A beta exposure stimulation in the primary cell culture is actually causing an acute inflammatory response in the primary microglia cells. So moving on, they wanted to investigate what are the metabolic differences or the metabolic output of such stimulation, and then they performed the C-course experiment. So for those of you that do not know what the C-course experiment is doing, so this is the readout that you get. So this is a machine that it senses the oxygen consumption in the medium, and from this experiment you add different blockers of the oxidative phosphorylation in order to test the maximum capacity and several other factors in the oxidative phosphorylation. And what is needed for now is that you have the oligomycin, that is, you record the basal respiration, the basal oxygen consumption from the medium. You add oligomycin, you block the complex 4, and then you should expect a decrease in the oxidative phosphorylation because you actually don't have enough proton gradient accumulating in the membrane of the mitochondria. Then you add the FCCP. The FCCP is a coupler, or it's a decoupling of the ATP pump, the complex 5, and this causes all the proton gradient that is stored in the intramembrane space to go out, and therefore allow the ATP synthase to produce ATP and consume oxygen. That's why you see that the oxygen consumption normally goes up. And finally, the adrotin and antimycin, which are inhibitors of complex 1 and complex 3, respectively, and then they see the complete drop because now all the complexes are blocked, so there are no more proton gradient accumulating in the membrane space. Sorry. So what you see that it's in the vehicle control. There is the maximal respiration goes up, so this is the highest capacity of oxidative phosphorylation in the cell. And then when you have the exposure to the LPS or the different forms of the A beta, it's actually this maximal respiration is going down. That means that the cells are not using anymore the oxidative phosphorylation in order to produce energy. So in addition to this, they stain for the mitochondria. So this is the microglia culture with the mitotracted deep red that stains the total mitochondria. So here you see actually the mitochondrial network inside, and in the first picture you see the vehicle treatment. You have these tubular-like structures, which are the normally looking mitochondria. They are fused mitochondria, so they are more prone to oxidative phosphorylation. And then you have this fission that is called that these tubular structures are actually becoming more dotted, and becoming a bit dot-like, and therefore you have a not efficient mitochondrial respiration. So you see that upon exposure to the oligomeric A beta, you actually get this kind of dysfunctional mitochondria. So the mitochondria are dysfunctioning, oxidative phosphorylation is down, so the cells need to produce energy, and therefore they turn glycolytic. They turn to the aerobic glycolysis, and as you see here, this is from the same machine, the C-Force machine, that they are. It's actually measuring now the extracellular acidification rate, and this is basically a pH probe that senses the differences in the pH of the medium. With glycolysis, there is lactic acid produced, and therefore the medium is getting acidified by time when the cells are glycolytic. Therefore, what you see here is that in baseline conditions, the vehicle, there is not much happening actually, because the cells are using oxidative phosphorylation, so they don't produce lactic acid. However, upon activation with either the A beta peptides or the LPS, so this inflammatory state of the primary microglia, they turn to glycolytic, they drop oxidative phosphorylation, and they increase glycolysis. And therefore, you see that over 6 hours, there is a high acidification of the medium. That means that the cells have shifted to a glycolytic metabolism. As a proof of concept here, they also measured the lactic acid in the medium, and you see that in all groups it's upregulated. Now, they hypothesized at this point that this shift from oxidative phosphorylation to glycolysis is what drives the pro-inflammatory phenotype of the microglia cells, and therefore they did a time course of the expression of the pro-inflammatory or the pro-interleukin-1 beta, and as you can see, it starts appearing at around 1 to 2 hours after the stimulus introduction to the medium, and is where you see here the 2 hours is most where you actually start to see the peak of glycolysis, and then they more or less see a plateau of the glycolytic rate. And following these 2 hours, you see that gradually the pro-interleukin beta is actually expressed, or its increased expression during time course until the 24 hours. In order to actually prove their point on the glycolytic shift and the pro-inflammatory phenotype, they pre-treated the cells. So before the exposure of the to the different A beta peptides or with 2DG. 2DG is the oxyglucose, which is an analog of glucose that gets internalized in the cells. It is recognized as glucose, but it cannot pass the second step of the glycolysis, the phosphorylation that's happening there, and therefore it just accumulates into the cell, blocking the glycolysis pathway. And as you can very well see, is that this pretreatment of 2DG in all different forms of the A beta peptide leads to a decrease in the RNA expression of interleukin beta or TNF alpha, suggesting that it blocks this pro-inflammatory shift of the primary microglia cells. Good. Now moving on, they wanted to find what is the pathway that is responsible for this metabolic reprogramming in the primary microglia cells, and they focus more on the mTOR pathway, basically because it's one of the pathways that pop up whenever there is something related with energy, with the energy status of the cell. And so the pathway with that they checked was the AKT-mTOR HIF1 alpha pathway. AKT is a coupler of the extracellular signals that lead to differences in energy balance. Then it gets phosphorylated, and in turn phosphorylate mTOR, which actually senses the energy status of the cell. And mTOR, this phosphorylated mTOR, while activated, will in turn phosphorylate P70S6 and P4ABP1. So this was just confirmation of the cascade that is activated, and the most important for their story is the HIF1 alpha transcription pathway. It's actually upregulated upon exposure to the LPS or the A beta peptides. GAPPH, which is an enzyme for a glycolytic enzyme and responsible for one of the steps of glycolysis, is actually getting as well upregulated with this with the treatment of LPS or the different A beta forms, and basically because GAPPH is controlled by the HIF1 alpha transcription factor. Good. So they to prove again their point, they wanted to block this pathway of the AKT-mTOR pathway. I forgot to mention before that they, as a negative control, they had the MMPK. MMPK is sensing the AMP and the ADP in the cell, and basically it senses the metabolic exhaustion and the energy exhaustion in the cell. Therefore, once the phosphorylated MMPK is down, it means that the mTOR pathway is getting high, is getting upregulated. So in order to prove their point, they treated the cells with rapamycin, which is an inhibitor of mTOR, prior to their exposure to the LPS, to the A beta forms. And what you see is that again the pro-inflammatory genes interleukin 1 beta and TNF alpha fail to increase significantly when the mTOR signaling is blocked. They used another inhibitor, metformin, which inhibits mTOR and increases the MMPK phosphorylation, and again they find the same result. So this pro-inflammatory status of the microglia cells is actually diminished, suggesting that the metabolic reprogramming of microglia that drives them to become pro-inflammatory due to the exposure to the A beta peptide is dependent on the mTOR pathway. Then they move on in an in vitro model of chronic treatment with A beta, and what they do in this setup, they have their primary microglia culture that they treat with the vehicle, two groups treat with the vehicle, and then one with the oligomeric A beta for 24 hours. And then they wash the cells and replenish the medium with a normal primary microglia medium without any stimuli, neither the vehicle nor the oligomeric A beta. they let the cells rest for 3 to 5 days, and then they're re-exposed to a second phase of drugs. So either they use only vehicle in order to have a control sample, either then they do this acute phase where they, for the first time, introduce the oligomeric A beta, and in the third group they just add again as a second hit oligomeric A beta, in order to mimic a chronic exposure to the stimulus. So with this experiment, what they see, their readouts again, is this pro-inflammatory response of the microglia. They see that the pro-IL-1 beta in the acute phase, so in the second group that they only sees for the first time the oligomeric A beta, the response of the production of the pro-interleukin-1 beta is actually increased, but in the chronic phase they don't see this increase, and this is also reflected in the RNA level of interleukin-1 beta and TNF alpha, and of course, as they saw before, there is no difference in the anti-inflammatory interleukin-10 and the interleukin-1 receptor alpha. In addition, with their phagocytic assay, they see that only after the acute phase of the exposure to the oligomeric A beta, they see the increase in the phagocytic capacity of the cells, while the chronic group is actually reaching again the control. So following this, they want to check the mTOR pathway, and then, as they did before, they check the phosphorylation of the AKT, mTOR, and the activation of the HIF1 alpha, and they see that only the acute phase is actually increasing highly significantly this response, while the chronic treatment, the chronic exposure to the A beta only partially increases the phosphorylation of AKT and mTOR and activation of HIF1 alpha, and partially activating this pathway, not as efficiently as in the acute phase. Glycolysis, measured now by the lactate release in the medium, is again increased only after the acute phase, while this chronic phase is similar to the vehicle control. So now we've seen before that the oxidative phosphorylation is getting down when you get exposure to this pro-inflammatory stimulus, the A beta exposure, and glycolysis is getting up. So now in this chronic model, we don't have the glycolysis to increase, so they just speculated that it shifts back to the oxidative phosphorylation. They don't respond anymore to the oligomeric A beta. But what they found from this OCR, the oxygen consumption rate again, was that the maximum respiration of the cells is not getting increased, as you see here in the bar plots, that it's still remaining quite low, suggesting that the cells are actually having a metabolic defect at this point of the chronic treatment of the A beta, and this functionally seems to impair the microglia cells. Now, there is some increase in necrosis also in the chronic phase. So this is the LDH release in the medium, that is an indicator of a necrosis. However, there are not so many apoptotic cells, or none detected at least. Okay, so having this in mind, they wanted to go in an in vivo model now and try to address this chronic response to the oligomeric A beta. And they now used wild-type mice and the 5x FAD line as a model of Alzheimer's disease, where you have deposition of A beta into the brain and gradually accumulation of the A beta plaques, and leading eventually to a severe disease burden. So they have these two mouse models, and then they inject into the lateral ventricle either PBS or the oligomeric A beta for 24 hours, and then they collect the microglia cells from the brain, excluding the olfactory bulb and cerebellum, and they perform an RNA sequencing. So the idea with this is that the wild-type mice see for the first time the oligomeric A beta, while the 5x FAD are already being exposed to A beta peptides, and therefore the microglia will see a second time, or they will see again oligomeric A beta. Of course, the oligomeric A beta is so the A beta exposure is still in the animal, so the 5 times FAD is always producing the excess A beta. So what they see here, this is the comparison between the wild type mice before and after the so with PBS or with oligomeric A beta treatment, and they see around 370 genes to be differentially upregulated, and all this in a GO term of biological processes. They see increased response, defense response, immune response, and generally response to stimuli. On the contrary, when they are looking at the differences between the PBS and oligomeric A beta injections in the 5 times FAD, they see far less genes. They see around 180 genes, and surprisingly for them, they see an overlapping in the biological processes that they are enriched with the GO terms, but of course the increase is far less than the one in the wild type mice. So in general, with this experiment, what they did here is a bit more detailed analysis of the different biological processes. So you see that the wild type mice respond more robustly to the oligomeric A beta, while the 5 times FAD is responding quite low. And what they have as an outcome is that these microglia from the brains of the 5 times FAD mice are barely activated, and they are not responding as much as the ones in the wild type. They wanted them to see what makes the response different. So they saw they investigated. So in this what are the differentially expressed genes in the wild type? They are not found in the 5 times FAD after the treatment of the A beta, and they come with a lot of pathways that are related with metabolism. So their point with this was that the microglia from the wild type mice seem to be more metabolically immunologically activated after the A beta exposure than the ones in the 5 times FAD, suggesting this A beta tolerance and the metabolic defect in the 5 times FAD microglia. So then they wanted to try and address somehow in vivo the differences in the metabolism of the microglia in the Alzheimer model, but now they go in a bit more basic analysis, leaving the 5 times FAD a bit in the side for now. So they use the tissue laser ablation technique. So basically, what you do, you do in vivo imaging in the mice, and then you increase the laser power in order to destroy the tissue in a certain spot, and then you measure how fast the microglia are actually attracted to the point. So because there is a excitotoxicity at the point of the lesion, there's a lot of chemoattraction of the microglia, and therefore they should go under normal conditions to the injury site. So they hypothesized that this process is highly energy dependent, and therefore the microglia needs to become glycolytic in order to do so, because glycolysis is 10 to 100 times more efficient in producing energy than the oxidative phosphorylation, although in terms of actual ATP molecules, glycolysis produces less. However, this, because the process is quite fast, is used to activate the cells in order to produce energy in higher speed. So here they gave intravenously 2DG for 3 hours before they do the laser ablation in the tissue, and basically what they saw is that under normal conditions with the PBS injected, after 1 hour you have the microglia reaching, or at least the branches of the microglia reaching to the point of the injury, and while there is no difference in the 2DG, actually it seems that they just don't care for the laser ablation at the point there. So there is no recruitment of the microglia and no turn of the microglia towards that point. They provide a supplemental video on the homeostatic motility of the microglia that is not affected by the 2DG injection, and therefore they make their point a bit stronger, saying that under normal conditions, microglia are not glycolytic, and therefore the 2DG doesn't have an effect on this motility. However, when they actually become activated and they need to produce a lot of energy by using glucose, then this is when this 2DG is actually blocking this process. And so this was their main, so the initial hypothesis that the microglia needs to become glycolytic in order to have this migratory phenotype. So they went further, and they actually not further, but rather back to this intracerebral ventricular injection of oligomeric A beta in order to activate microglia and see whether this makes the microglia first more responsive to this laser ablation. From their in vitro experiments, they expect the microglia, upon exposure to the oligomeric A beta, to become more glycolytic. And therefore, they do this 24 hours before their laser ablation, and they found a small but rather significant increase in the speed of the microglia response after exposure to the oligomeric A beta, comparing to the PBS injection, suggesting that this boost that they got due to their activation is actually beneficial to the migratory phenotype of the microglia to the ablation point. They went then to the 5 times FAD model, and they did similar analysis, and therefore they did the tissue ablation in both groups, the wild type and the 5 times FAD. And what they see is that in the 5 times FAD, the microglia is far slower and unresponsive to the laser ablation compared to the wild types. And for this, they hypothesize that this is probably due to this decreased in microglial activity and glycolytic capacity that is actually not responding as fast. Now, one of the points that it was raised, and possibly by the reviewers in this case, was that from the Karen Saul paper from the Ido Ahmet group in 2017, the plaque-associated microglia is downregulated one receptor, the P2R-Y12, cholinergic receptor, which is basically an chemoattractant receptor, and it's very much involved in the chemoattraction in such models of tissue ablation. And therefore, the question that arose was whether, so if you have microglia in the 5 times FAD model that downregulate this receptor, then you should expect this decrease in the response to the tissue ablation, but and it's not due to this metabolic exhaustion, metabolic tolerance, this A beta tolerance for the microglia. They provided data from their RNA sequencing. This, of course, not plaque-associated microglia, but whole tissue microglia, and they see no significant decrease in the levels of the P2R-Y12 among the different groups, and they claim that it's not due to the downregulation of the receptor, but rather that the in vivo microglia responses are dependent on the glycolytic metabolism and are defective in the AD model mice. Now, of course, here in the 5 times FAD mice, it's done in the cortex, the in vivo imaging, and the tissue ablation. However, we don't know whether there are plaques near this area, so you might have some plaque-associated microglia being activated, or at least sensing this kind of tissue ablation. But this is something that they don't address. So then the next question, or not question, but rather hypothesis that they had, is that if they boost the metabolic capacity of the glycolytic metabolic capacity of microglia in the 5 times FAD model, they would rescue this diminished response that the microglia have, or this tolerance to the A beta. They thought to use interferon gamma as a metabolic booster for glycolysis in the microglia for 2 reasons. First of all, because it's been shown to activate glycolysis in innate immune cells, and secondly, because it's a potent activator of the mTOR. So they went back to their in vitro setup and they treated the oligomeric A beta in addition with their primary microglia cells with oligomeric A beta, in addition with interferon gamma. And what they see is that they actually increasing the glycolytic capacity of the cells, and suggesting, or at least how they suggest, that the interferon gamma is actually boosting the glycolytic metabolism of the microglia. They use then the data from the virus to show that the interferon gamma receptor is actually expressed mainly in microglia cells, and other cells are not that potent expressions of this receptor. So in general, what they claim is that the interferon gamma is able to restore this inflammatory response to A beta by upregulating the glycolysis. Now, one missing control from this experiment is what happens if you just give interferon gamma to the cells and without any exposure to the oligomeric A beta. You should expect an increase in glycolysis either way. So my question here is whether what you see here is just a masking effect of the and the oligomeric A beta doesn't really play a role, but rather the interferon gamma just masks everything due to their potent activation in the cellular pathway. And I just did a brief search in the literature to see whether indeed it is the case. So if you treat bone marrow derived macrophages with interferon gamma only, what you see is that the extracellular acidification is actually increasing by itself. So this is something that misses from the paper, at least in my point of view. And so then they went again to this chronic model in order to see whether interferon gamma can actually rescue this metabolic defects of the microglia in the chronic in vitro model of A beta exposure. And they used only now the chronic phase, and what they did is that before they give the second hit of the oligomeric A beta, they treat the cells with interferon gamma for 1 hour. They went to the mTOR pathway. They see that there is in this tolerant microglia that has been boosted with interferon gamma before. There is an increase in the phosphorylation of PKT, phosphorylation of the mTOR, and expression of HIF1 alpha, which eventually leads to an increased glycolysis. So rescuing this this metabolic exhaustion of the cells that they suggested before in the acute versus chronic phase. So this was also translated to an increase in the TNF alpha expression and increased in internalization of the microspheres, suggesting again this rescue in the microglia response. Of course, here again, what is missing for me is what happens if you just give interferon gamma in an acute phase of oligomeric A beta. Would you expect the same response? And it's rather again a masking phenotype of interferon gamma, not the second exposure of the oligomeric A beta. So then these in vitro data were convincing enough for them to continue and in vivo model, and now try to see whether administration of interferon gamma systemically before the laser ablation and in vivo imaging is actually affecting the microglia response towards this tissue ablation. And basically what they see is that comparing the 5 times FAD now with the injected ones, with interferon gamma, that there is a rescue in the response, and they are actually faster in responding to this tissue ablation, tissue laser ablation. Now this is all sterile injuries in the brain with laser ablation, and then they wanted to see actually in the disease whether this has an effect. So they continue with the in vivo imaging model, and I think I lost my sensor somewhere. But anyway, you see, here it is. Sorry. So they treated the mice. Now this is a 5 days in vivo imaging of the same mouse. So they injected before the mice with metoxy. Metoxy is a dye that you inject in the peritoneally, and then crosses the blood-brain barrier and stains the A beta deposition. So here you see the plaques stained with metoxy. So they injected before the acquisition. Then for 2 days they were imaging the mice, and then in day 0 they gave interferon gamma. They gave again metoxy because every 2 days they injected metoxy to keep the plaque stain. And afterwards they imaged again the brains of the mice, and what they see in general is that the volume of the GFP signal on the fluoxetine GFP around the plaques is actually increasing, suggesting that more microglia cells are actually accumulating around the plaques. And having done this short period experiment, they then went further and asked, okay, what happens if we give interferon gamma for a longer time period? Can this affect the disease burden? And they treated the mice from age of 4.5 weeks to age of 7.5 weeks, so for 3 months, with interferon gamma IP, given once a week a dose. And then post-mortem, they analyzed the brains. They stained for the A beta deposition with 48 antibody that stained A beta, and then measured how many microglia cells are around the plaques. And again, here they see that indeed after this 3-month period, you have higher number of IBA1 positive cells surrounding the plaque. And not only this, but also that these microglia cells have more A beta internalized, suggesting an increased phagocytic activity. On the other hand, the A beta depositions in the brain is actually diminished, suggesting that the clearance of the A beta is faster or more potent in the interferon gamma injected mice. In order to prove that this is not just an artifact from the production of A beta, they checked the APP and the PS1 production. So this is staying the same, which means that the production of the A beta is not altered from the neurons, is not altered, but rather that the microglia cells are more and they phagocyte more efficiently the A beta. So they tested, they checked also whether there are some changes in the synaptic density, and there they didn't see any significant increase. However, what they found is that the injection of interferon gamma is actually rescuing or at least increases the neuronal survival in the 5 times bug mice. Finally, in order to have a more behavioral readout and to see whether this interferon gamma boosting of metabolism in the microglia is actually affecting the behavior of the mice and alleviates a bit the disease burden, they did a Y-maze test, and then they see that in the end the injections with interferon gamma seem to be beneficial in the response of the mice in this test. So wrapping up, what are the main points and the highlights of this paper is that the A beta seems to induce a metabolic shift of microglia from deoxyribonucleic phosphorylation to glycolysis, and that is an mTOR HIF1 alpha dependent pathway. On the other hand, the chronic exposure of the A beta induces metabolic defects in microglia cell, and interferon gamma can actually rescue this this immunological function of the microglia. And with this, I would like to thank you for your attention. I'm open to your questions. Until you type or think of my questions, I will tell you my idea about this paper. And so I find some aspects quite nice that they do. However, I find that a lot of the metabolic stuff are done in the in vitro system, and not a lot of metabolic readouts have been performed in vivo. So there is, of course, a big gap between the in vitro flow system and the in vivo response. There are many cells there. There are a lot of different changes in the milieu of the brain, and therefore this, for me, it's a disadvantage of the paper. In addition, giving a beta peptide or something that the microglia can phagocyte will definitely increase their metabolic, would change from oxidative phosphorylation to glycolysis. Basically, this is a feature that any cell, so any immune cell and even microglia that is going to get activated will change to the glycolytic pathway. For example, if you treat the primary microglia cell culture with myelin debris, you'll see a similar effect, that they turn glycolytic and they get this activation of the mTOR pathway. So this, again, it's not very specific to the A-beta, but rather it's a more generalized response to an activation of the microglia cells. And all in all, what for me it's not that catchy is having a title saying the breakdown of metabolic reprogramming causes a microglia dysfunction in Alzheimer's disease, but in the end, what you saw in terms of metabolism is basically in vitro exposure to A-beta, and not that much translated to the in vivo model of the 5x findings. So that was my idea about the paper. I'm open for your questions.
Yeah, thank you so much, Nikos. And guys, yeah, you can simply type the questions in the Q&A section. So just feel free to write, and yeah, also let me know if you think this is a good option for receiving the questions. And I tried to open it also beforehand, that you can write the questions early on. I think during the webinar I will open it from the beginning on, so that people can already have written a lot of questions that we can eventually choose from. Yeah, so there's already one question. I guess Nikos can already.
Yes. So this is a very interesting question. They've never showed wild-type mice injected with interferon in either of their tests. In general, what I would believe, because it's a pro-inflammatory stimulus, is that the mice will already have a phenotype. It might be similar, or at least resembling a bit an LPS injection. It's also quite long, so you have this 3 months exposure to interferon gamma, but in the case of the paper, they never saw that. What happens in the wild-type mice with interferon gamma. And also in terms of the motility of the cells, for example, they use the 5x FAD model. They inject
this is a very interferon gamma,
and after laser ablation they see this rescue. However, in the wild type they never inject interferon gamma. So I can see the question. I don't know.
I guess I will double check it, but I think
this is so old question. You only saw your own question. Because I have so old questions, and this is because I'm a presenter. If you go as a guest.
Yeah, maybe we check. Can you log in? Would you like to yeah, Monique, I will double check that, but yes, exactly. Yeah, I will. I just thought about it, that I will read the question out loud, and then I'll address it to Nico. So basically, Subra's question was, how do wild-type mice behave after treating with interferon gamma? And as Nikos mentioned, it's a really interesting question. I guess you heard. I do not read.
Yeah, they
They
didn't
fit
the mice. They don't show any data on this. It would be interesting to see if they also failed to do this in the laser ablation test, and so they never treated wild type mice with reprogramming and see whether the motility of microglia is actually changed, or the recruitment to the laser site.
Yeah. So do you guys have any more questions? It would be nice. Just ask anything that we.
I hope
I
there
hope
was
there was
first of all was everything smooth with the presentation itself. Do you experience any lags? Did you hear me well?
Any technicalities, or if you think maybe something in the layout can be fit better, we will anyway broadcast it then in the room. Yeah, it's just because we are in the same room right now, so there is you hear Nikos twice probably. But we just wanted to test the technicalities, and during the webinar, hopefully then by broadcasting, things will be also a lot smoother. But also in case you cannot attend in the rooms that are booked in the either cities, you can basically open it from your laptop, and yeah. So if everything is functioning, then it's all right. So I don't know. I give you one more minute. If you want to address any more questions to Nikos, then just feel free. And if not, then I guess that's it. Yeah, thank you for attending. And I think we had a late joiner. Stefan just joined recently. So I will make a YouTube channel, and I will upload the videos. For now privately, and I will send you the link in case anyone wants to recheck the webinar from the people who did not attend it. Okay, so there seem to be no more questions.
Thanks a lot
yeah.
for your patience. I hope everything is fine. And the actual seminar is on the 15th, right?
Yeah, so it's on the 15th, and it is at 5 p.m. if I remember correctly. And as Christine also mentioned, there will be a room in Berlin. I don't know about Israel, but also here in Freiburg we booked the conference room. So
in the Narvik Center.
Yeah, exactly. It's exactly. So if you would like to attend it together, and then we can basically watch it as one, or you can also simply watch it from your laptops. But it will be nicer if you come, so that I don't know, we have a crowd. Yeah. So thank you all so much, and I would like also to thank Nikos again for being the sacrificed sheep.
Yeah, it's always nice.
Outside, right?
Yeah. And yeah, I wish you all a good evening. Bye bye, guys.
Ciao.